On this slide, we can see a picture of, again, half of a replication bubble. So we're looking at the point of origin right here and here on the old DNA, and then the right-hand side of the replication bubble. And here's the DNA that is still in the double-stranded form outside of the replication bubble. The two strands of old DNA, remember, are oriented anti-parallel to each other. And that means that the new DNA will have to be made in two different directions, because the DNA polymerase enzyme can only add new nucleotides to the 3' end. So lots and lots of 3's and 5's in this picture. Let's take a look. Here's an old strand of DNA in gray. Here's its 3' end. So that means this is its 5' end over here. They're showing us its actual 5' end over here. The other strand of old DNA is anti-parallel to that. So it has a 5' here and a 3' here. Now, when this enzyme makes new DNA, the new DNA has to be anti-parallel to the old DNA. So if there's a 3' in the old, right here, that means there has to be a 5' in the new, right here. Same thing on the other strand. If the 5' end is here on the old, then the 3' end has to be here on the new. The enzyme that's building the new DNA can only add new nucleotides to the 3' end. The 3' end is right here, where the arrowhead is, on this new DNA, toward the replication fork. The 3' end with the arrowhead is here on this strand, toward the origin of replication. That means this new DNA is being made using leading strand synthesis. This new DNA is being made using lagging strand synthesis. If I were to come back in a second or 2 seconds, what you would see is this. The replication bubble has grown. It has continued to open up. So where the fork used to be sitting here, it's now sitting here. The new DNA that's being made using leading strand synthesis is longer, and it's one continuous piece. Remember, we're adding new bases at this arrow, at this 3' end. And this is right at the fork. So as the fork moves this way, the new DNA just gets longer and longer and longer in one continuous piece. Now look at the place where the leading strand is, where the lagging strand synthesis is going on. Here we saw it growing at time number 1. Here it is at time number 2. It's also gotten longer. The replication bubble is opening up, but on this strand, on this area where lagging strand synthesis is taking place, it's gotten longer over here toward the point of origin. So examine exactly what has happened. I've got this. Yikes. I've got this whole area of old DNA that hasn't been copied yet. We're building this new DNA this way. How are we going to deal with this? How is the cell going to get this copied? Well, the answer is the enzyme has to jump back. It has to jump back toward the ever-opening replication fork and start making new DNA. So it's made this piece, this fragment. Now it needs to jump back and start making a new fragment. Lagging strand synthesis. Now, DNA polymerase is an amazing enzyme. It can do multiple jobs for a cell, which is pretty remarkable. But it has to have a couple of things in place for it to do its work building DNA. Number one, it has to have a single strand of old DNA. It has to have a single-stranded template of old DNA. And it has to have a 3' hydroxy functional group to add a new nucleotide to. It has to have that 3' hydroxy group sort of exposed so it can bring in a DNTP and add it right there. Build a phosphodiester bond right there. So, according to those needs, we've got a little bit of a problem. Because when you open up a replication bubble in old DNA, there is no place that has an exposed 3' hydroxy group. There is just nothing for the enzyme to add a nucleotide to. Now, the way the cell gets around that problem is by using what is called a primer. The cell is going to build a primer on the old DNA to get DNA replication started. And what a primer is actually a short piece of RNA, not DNA. And it will be covalently built and then attached in a complementary way to the existing old DNA. There's an enzyme that does this work, of course there is, and it's called Primase. Again, those are good names for enzymes, the ones that basically tell us what they do. So the primer of RNA will be built by Primase right onto the old DNA. That primer will have an exposed 3' hydroxy functional group on it. The DNA polymerase enzyme will come in, it will see that 3' group, and it will start adding new nucleotides, new DNTPs, one after another, using the old DNA as a template. On the slide we looked at a moment ago, you can actually see the RNA primer because it's in a different color. So this yellow region right here represents the RNA primer. Down here, this yellow region represents the RNA primer. So you can imagine that once this whole piece of old DNA has been replicated, we have another problem to deal with. We have all of these little primers that are made out of RNA, not DNA, and they need to be dealt with. So after the new DNA is made, all of these RNA primers have to be removed and replaced with DNA. Now, here's a question. Why is it that you think primers are made out of RNA? Why doesn't the cell just make a primer out of DNA? It would be so much easier. You could place a little primer of DNA and get the enzyme started, and then you wouldn't need to take them out because they would already be made out of DNA. Well, the answer to this question relates to evolution. If you recall, we believe that the first form of nucleic acid that existed on our planet was actually RNA and not DNA. The very first sort of primitive life form on planet Earth is believed to have been a self-replicating RNA molecule. So the idea is that RNA was the original genetic material on planet Earth, and that eventually DNA evolved out of RNA, if you will. DNA makes a better molecule to store your genetic information in, and that's because DNA is a more stable molecule. But what likely has happened is that the RNA origins of nucleic acid have persisted through this little remnant right here. That the primer system using RNA was in place first, and it was maintained. And it was maintained because it works well. That's usually why something persists, evolutionarily speaking. It works well. The RNA primer has persisted. On this slide, we can see an RNA primer being placed on an old strand of DNA. So here's our old strand with individual bases coming off of it. Here's the RNA primer. RNA is complementary to DNA. It's the whole basis of the process of transcription. Where there's a G in the old DNA, we can build a C in RNA. That looks exactly the same as the complementary base pairing we see in DNA. Here's where it gets a little different. Where there's an A in DNA, we would normally think about placing a T, but there is no T in RNA, recall. The T is replaced with a U, which stands for uracil. So the RNA will be complementary matched with the DNA, and this short little piece of RNA primer will be laid down. It provides this. It provides on its 3' end a hydroxy group, a functional group. The DNA polymerase enzyme can come in if it sees this. It can work with this. It can attach a DNTP to this end right here because of that hydroxy group. So once that primer is in place, the DNA polymerase enzyme is going to come in and add DNTPs to the 3' hydroxy end of the primer. And the DNTPs will be added one after another, after another, after another, according to the template of old DNA. DNA polymerase is a fascinating enzyme, and like all big proteins, it has a particular shape to it, and that shape relates directly to its function. When you look at the three-dimensional model of DNA polymerase, it's fascinating because it looks almost like a hand that grips onto that DNA template strand, just like a hand grabbing onto a rope or a pipe. The catalysis, the enzymatic action of DNA polymerase, the place where a phosphodiester bond is being built, is right in the groove of the hand, between the index finger and the thumb. Now, one of the issues with the enzyme is this. Remember, I told you that DNA polymerase, once it finishes building a new strand of DNA, it can then proofread that strand and find any errors that it might have made and fix them. In order to do that proofreading, that enzyme has to be able to move backwards along the new strand of DNA. So it builds the new strand in one direction, and then when it gets to the end, when it finishes building it, it literally starts to move backwards along that strand to proofread it. What that means is that that enzyme is a little bit slippery. It can go in both directions. And the cell needs to step in and prevent that from happening during the process of DNA replication. So here's where another one of these accessory proteins comes in. There's a protein that sits right behind the DNA polymerase enzyme. It's called the sliding clamp. It functions almost like a nut on a screw. It sits behind, if you will, the DNA polymerase enzyme, and it prevents it from backsliding. This is a busy looking drawing, but it shows us some of these enzymes and accessory proteins that we've been talking about so far. Now, right now we're discussing the leading strand form of DNA synthesis just because it's more straightforward. We'll get to the lagging strand form in just a minute. We're looking at the gray strands of old DNA right here. So again, this is half of a replication bubble. Here's the replication fork. Here's the DNA that's still double-stranded outside of the fork. Remember, the very first thing that happened was this helicase enzyme came in and opened up the double strand at the point of origin. Single-stranded DNA binding proteins came in, and we shorthand those SSBP. They came in, and they're going to sit on the single strands and keep them from snapping back together again and reforming the double helix. Primase, shown in yellow, is going to come in and it's going to build a primer. It's going to build a short stretch of RNA to get replication started. Topoisomerase is going to come in outside of the replication bubble and it's going to relieve tension. In comes the DNA polymerase enzyme, and that's shown in this lighter green color here. And here in the darker green color is the nut behind it, the sliding clamp. Since this is leading strand synthesis, it's going to start at the point of origin and it's going to head towards the open replication fork. So the enzyme is moving this way. They've shown us that the enzyme, the DNA polymerase enzyme, is literally sort of wrapped around. It's grabbing the old strand of DNA. See how this looks like almost like a thumb? And this would be the bottom of the hand, the fingers. It literally grabs onto that old strand like a hand would grab a rope. So the synthesis of the lagging strand regions is a little more complicated, and that's because the DNA is not being made continuously, it's being made in fragments. So these Okazaki fragments are made a little bit more slowly than one long continuous piece of DNA. And that's why this process gets named lagging strand. It lags behind a little bit because it just takes that much longer to build in fragments than to build continuously. Again, another busy slide, but we'll follow along together and see if we can identify all of these players. It's a little bit harder to see the old DNA in this slide, but here's where it's still double-stranded. Here's the replication fork. Here's one old strand, and here's the other old strand. Now what they've done is they've sort of ghosted what's happening up here. That's why this looks white, and the old strand looks very faint. This is leading strand synthesis going on over here. Down at the bottom, this is lagging strand synthesis. Here's the helicase that opened us up, single-stranded binding proteins that are holding us open, primase that's building us a primer, topoisomerase that's relieving pressure. Here's our enzyme. Here's the DNA polymerase, light green. Here's the sliding clamp, dark green. We're building DNA in this direction because this is lagging strand synthesis. We're building DNA towards the origin of replication. So the fork continues to open up. We build our first fragment, and then the enzyme has to let go and jump back towards the fork and start building a second fragment. Now that means we're going to need a second primer. So it starts at the 3' hydroxy end and it builds new DNA. Then the fork opens up even more. So the enzyme has to jump back again. It has to lay down, primase has to lay down another primer, and the enzyme makes new DNA. So we've built 3 fragments here. That means that those fragments have to be joined together somehow. And this brings in a couple of additional enzymes. First things first, RNA primers have to be removed from both the leading strand areas and the lagging strand areas. And there's an enzyme that does that. The primers are removed and replaced with DNA by a molecule called DNA Polymerase 1. Remember, all DNA polymerase enzymes are numbered. It's not critical that we remember the numbers, except in this case. It's DNA Polymerase 1 that removes primer and replaces it with DNA. The short fragments then of DNA will then be joined together with an enzyme called DNA ligase. So these are the two additional enzymes that are involved in lagging strand synthesis that are going to bring those Okazaki fragments together into one new strand of DNA. And that's what they're showing us on this slide. Here's double-stranded DNA. Here's the replication fork. Here's an old strand up top that's being replicated using leading strand synthesis. It's been ghosted. Can't really see it. Here's the other strand of old DNA. New DNA is being built on it using lagging strand synthesis. We've got fragments here. Here's DNA polymerase 1. And I love this drawing because they've drawn it to show that it's sort of chewing out the primer. Little pieces of primer are flying into the air in this drawing. And then new DNA is being replaced in here. DNA ligase comes in, and DNA ligase is going to seal that little gap right there between the fragments. I included this slide because it's a complete list of all of the enzymes and accessory proteins that are used during DNA replication in prokaryotic cells. They've divided this big table up to show us that some of these enzymes and proteins are involved in both leading and lagging strand synthesis. So helicase, single-stranded binding proteins, the DNA polymerase enzyme itself, topoisomerase enzyme. These are involved all the time. It doesn't matter whether you're doing leading or lagging strand synthesis. If you're doing leading strand, the list of other molecules is shorter than if you're doing lagging strand synthesis. To perform leading strand synthesis, you have to have a primase enzyme, you have to have DNA polymerase, and you have to have the sliding clamp. To perform lagging strand synthesis, you also have to have primase and DNA polymerase and the sliding clamp, but you need those two additional enzymes. You need DNA polymerase 1 and you need DNA ligase.